Hi everyone, today our video will cover all the important details about the topic of quorum sensing in bacteria. Quorum sensing refers to the process of bacterial communication. More specifically, it is a type of signaling pathway that is used by gram-negative and certain gram-positive bacteria to regulate gene expression. This method of communication is reliant on cell density. Gene regulation occurs as the bacteria release autoinducers, which are signaling molecules that increase in number with increase in cell density. Quorum sensing may be observed during production of antibiotics, formation of biofilms, virulence, motility, and more. Today, we will tell you about a specific group of bacteria known as Alivibrio fischeri, who use quorum sensing to colonize the gut of the Hawaiian bobtail squid symbiotically.
So the quorum sensing system in the relationship between Alivibrio fischeri and the bobtail squid is the most studied and has provided a model for understanding how quorum sensing pathways work. The bacteria and the squid live in a symbiotic relationship, both contributing to the eventual bioluminescence. Alivibrio fischeri is actually the only bacteria that's able to colonize the gut of the squid due to a toxic compound that the squid produces. During the day, while the squid is in hiding in the sand, the bacteria will colonize its gut, and then after the night, after it's done its preying, the squid will expel the bacteria from its gut since bioluminescence is no longer necessary. This relationship is beneficial for the bacteria as it's the only bacteria that's in the gut and has a bounty of nutrients available to it. After colonizing the gut, it even loses its flagella since it no longer needs it since it's where it needs to be. The relationship is also beneficial for the squid since it's hunting at night. The little squid's bioluminescent light organs make it look like it's a reflection of the moonlight, so it's camouflaged from predators. The bacteria even induces a morphological change in the epithelial cells of the light organs of the squid. It causes swelling of the cytoplasm so that the bacteria are forced together. The organ shrinks in size, causing the bacteria to pack closer together and increasing the cell density in the light organ and causing even brighter luminescence due to quorum sensing. An additional important component of the quorum sensing pathway, in addition to the Lux operon, is the AINES gene. The AINES gene produces the AINES protein, which has been very important for the early stages of colonization.
So now let's get into genetics of bioluminescence. The Lux operon encodes for the proteins needed for bioluminescence. The primary enzyme luciferase is what catalyzes the reaction that emits light. So we want the expression of the Lux A and B genes, which code for the luciferase enzyme. Lux I is responsible for making the autoinducers, also known as AHO. These molecules are constantly expressed in the Lux operon, and they're necessary because the Lux R generates the regulatory protein, and the autoinducers are then able to bind to the Lux R, the regulatory protein, which then stimulates the expression of the Lux operon because the complex with the regulatory protein and the autoinducer can then bind to the Lux box, which is located in the promoter, which then allows the RNA polymerase to come bind to the promoter and allow expression of the genes that are downstream of the promoter. And Lux A and B generate the luciferase enzyme, while Lux C, D, and E are necessary for the fatty acid reductase complex, which is part of the luciferase mechanism. We don't really care about Lux G because it's not important for the mechanism for luciferase.